Caffeine, or 1,3,7-trimethylxanthine, is a plant-derived alkaloid and psychostimulant that is naturally occurring in tea leaves, coffee, cocoa beans, and kola nuts. Caffeine is a mild central nervous system stimulant, temporarily warding off drowsiness and restoring alertness. A dose of 50 to 200 milligrams usually leads to mild stimulation. Individuals are exposed to caffeine via beverages, food, over-the-counter drugs, prescription drugs, herbals, or dietary supplements. The most likely route of exposure is oral intake. Caffeine is absorbed in the gastrointestinal tract. Approximately 80 to 100% of caffeine is absorbed across the gastrointestinal epithelium. Caffeine has a log POW of negative 0.07, making it slightly more hydrophilic than hydrophobic, but is well within the drug-like range of log POW values, allowing for simple diffusion across cell membranes. Caffeine contains two amide functional groups and two amine groups. The amide functional group has the highest polarity in the absence of any ionic functional groups, making it the greatest hindrance to simple transcellular diffusion. According to one source, the pKa of caffeine is 0.61, which is below pH values encountered in the GI tract, making caffeine deprotonated and neutral, facilitating simple diffusion across the gastrointestinal epithelium. After absorption across the GI epithelium, caffeine enters the liver via the hepatic portal vein before crossing the hepatic endothelium and vascular endothelium membranes and entering the heart to be distributed to the rest of the body. Caffeine is readily distributed into body tissues and readily crosses the placenta and blood-brain barrier. Peak plasma levels are achieved within 15 to 45 minutes after oral intake. Absorbed caffeine reaches maximum blood concentration within 1 to 1.5 hours. Most of the caffeine dose ends up as metabolites in the urine, therefore very little of the caffeine dose is bound to proteins or other molecules. However, due to its structural similarity to adenosine, caffeine acts as a competitive inhibitor by binding to adenosine receptors to exert its mechanism of toxicity on the sinoatrial node of the heart. Given the relative hydrophobic and neutral properties of caffeine, it is easily absorbed across most membranes. When considering distribution, the demethylation of caffeine to its metabolites increases hydrophilicity of the molecule. However, the metabolites are still hydrophobic enough to cross most barriers. Metabolism of caffeine occurs in the liver and begins with phase 1 demethylation. In adults, about 80% of a dose of caffeine is metabolized in the liver to 1,7-dimethylxanthine, also known as paraxanthine, making it the main metabolic pathway. Demethylation of caffeine to 1,7-dimethylxanthine involves the removal of a methyl group from the third nitrogen and has the effect of increasing lipolysis, leading to elevated glycerol and free fatty acid levels in the blood plasma. About 10% of caffeine is metabolized to 3,7-dimethylxanthine, or also known as theobromine, which is responsible for dilation of blood vessels and increases urine volume. Theobromine is also the principal alkaloid in the cocoa bean and therefore chocolate. About 4% of caffeine is metabolized to 1,3-dimethylxanthine, also known as theophylline, which relaxes the smooth muscles of the bronchi and used to treat asthma. The therapeutic dose of theophylline, however, is many times greater than the levels attained from caffeine metabolism. These compounds are further demethylated to monomethylxanthines and then undergo phase 1 hydroxylation to form uric acid. The metabolism of caffeine into its metabolites increases hydrophilicity, hindering distribution and facilitating excretion in the urine. The metabolism of caffeine is catalyzed by cytochrome P450 isozyme 1A2. Cytochrome P450 belongs to the family of monooxygenases, which is a group of enzymes that catalyze the oxidation of organic substances. Cytochrome P450 plays a central role in the oxidative metabolism of a large number of compounds and has broad specificity. Cytochrome P450 is most prominent in hepatic tissues. Some people metabolize caffeine more slowly than the general population due to variations in a specific cytochrome P450 gene. After caffeine is metabolized in the liver to paraxanthine, theobromine, and theophylline, these compounds are further demethylated to monomethylxanthines, which then undergo hydroxylation to uric acids in order to be excreted. Caffeine and its metabolites are excreted mainly by the kidneys, where it is filtered into the glomeruli and remains in the tubular lumen to be excreted with urine. Uric acid is secreted by a unique organic anion transporter, URAT1, in the renal tubules. During elimination, approximately 85% of caffeine metabolites are excreted in the urine, with approximately 1% being excreted as unchanged caffeine. In the first-pass metabolism of caffeine in the liver, caffeine exerts its toxicity through its metabolites. After oral intake, approximately 8% of caffeine is demethylated at the 3 position to form paraxanthine, making it the main metabolite of caffeine. Paraxanthine has the effect of increasing lipolysis, leading to elevated glycerol and free fatty acid levels in the blood plasma. As well, paraxanthine acts as a competitive non-selective phosphodiesterase inhibitor and a non-selective adenosine receptor antagonist in the heart. Caffeine has many targets, including the brain, but this project focuses on the sinoatrial node of the heart. The sinoatrial node is a small mass of specialized cardiac muscle fibers. It is located in the right atrium of the heart and acts as a pacemaker of the cardiac conduction system by generating at regular intervals the electrical impulses that generate the rhythmic contraction of the heart. Pacemaker activity is regulated by the depolarization of cell membranes due to inward ion currents across the membrane, including the hyperpolarization-activated funny current or pacemaker current, which generates action potentials. Studies show that the pacemaking mechanism is also mediated by a calcium clock. In the sinoatrial node cells, calcium activates adenylate cyclase, which in turn activates cyclic adenosine monophosphate. Cyclic AMP mediates protein kinase A, which phosphorylates calcium cycle proteins. This results in a release of calcium from the sarcoplasmic reticulum through ryanodine receptors, generating rhythmic calcium oscillations during diastolic depolarization, triggering the generation of rhythmic action potentials in the cell membrane. This phenomenon is called calcium-induced calcium release and increases the myoplasmic free calcium concentration caused by muscle contractions. Caffeine directly modifies the contractility of the musculature in the heart and blood vessels. Potential toxic effects of caffeine on the sinoatrial node can range from a moderate increase in heart rate to more severe cardiac arrhythmias. Caffeine and its primary metabolites are structurally similar to adenosine. Paraxanthine in particular acts as a non-selective adenosine receptor antagonist in the heart by preventing the binding of adenosine. This subsequently inhibits the release of G proteins, which normal function is to inhibit adenylate cyclase. This causes adenylate cyclase to activate cyclic AMP and protein kinase A, which creates an increase of myoplasmic calcium release from the sarcoplasmic reticulum through ryanodine receptors. Increases in levels of protein kinase A also activates the RAS signaling system, leading to activation of MAPK pathways, which promotes hypertrophy and cell apoptosis. Xanthine also inhibits phosphodiesterase, which are normally responsible for the breakdown of cyclic AMP. Therefore, this leads to an overall unmediated increase in cyclic AMP and ultimately an influx of calcium. In a positive feed-forward loop, higher concentrations of calcium further activates pKa, leading to the release of more caffeine. By increasing the amount of caffeine present within the sinoatrial node, there is an increase in the generation of action potentials and therefore heart rate. Severe cases of caffeine overdose can cause cardiac arrhythmias leading to death. Increases in calcium leads to production of superoxide ion, promoting apoptosis. It can activate nitric oxide synthase, generating the nitric oxide radical. These reactive oxygen species can react to form peroxynitrite, causing DNA damage. They can also induce lipid peroxidation. Caffeine is a mild central nervous system stimulant, temporarily warding off drowsiness, increasing attention and memory, increasing heart rate, and restoring alertness. At sufficient doses, caffeine can cause flushing, chills, agitation, irritability, loss of appetite, weakness, and tremors. In the case of overdose, which is rare in adults and requires ingestion of a dose typically in excess of 5 grams, caffeine can cause hypertension, hypotension, tachycardia, heart arrhythmias, and cardiac arrest caused by the mechanism of toxicity on the sinoatrial node previously outlined. In severe cases, overdose of caffeine can cause death. Low doses of caffeine can have therapeutic effects on the body. Caffeine can be used to treat pain as well as respiratory problems such as asthma and apnea.